Hi everyone, in this video we are going to discuss about structure and function of polymerase enzyme in prokaryotes. So in prokaryotes there are 5 polymerases are present. So there are 5 polymerases present in prokaryotes. These polymerases are called polymerase 1, 2, 3, 4 and 5. So coming to their structure, so when we look into the structure of the polymerase enzyme, so in polymerase see these green structures are the polymerases. So in these polymerases what we can see, so majorly three domains are present in these. So they're called thumb domain, finger domain and farm domain. So these three domains are present, and so these polymerases basically what they do in a replication, so they synthesize the daughter DNA using template DNA. So they adds complementary nucleotides to the template DNA, so that they synthesizes the daughter DNA. So apart from this, so polymerases not only involved in the synthesis of the DNA, also these polymerases they perform the editing functions. So they synthesize the DNA, so that means they replicate the DNA. In addition to that, also they repair the DNA. So when they repair the DNA, so when at the 3' end of the this growing nucleotide, this growing daughter DNA, wrong nucleotides are incorporated. So these wrong nucleotides are removed by these polymerases. So that's why we say that these polymerases have exonuclease activity, since they remove these nucleotides at the ends of the growing DNA. So that means, so they perform the function of the replication by synthesizing the daughter DNA. Also they repair the DNA when wrong nucleotides are incorporated. So apart from this polymerases also have other function, that is called removal function. So they remove the RNA primer. So you know that, so in a DNA replication in prokaryotes, where the polymerases can't initiate the DNA replication because they need a primer which is a RNA primer, so that at the end of this RNA primer, so the DNA polymerases can add the nucleotides to the daughter DNA strand. So that's why, so these polymerases have three functions. One is a removal function. So when DNA replication is completed, so these RNA primers are removed by the same polymerase enzymes only. So they have a removal function, they have a replication function, that means that they synthesize the daughter DNA. Also they repair the DNA by removing the wrong nucleotides at the 3' end. Okay, when we look into the polymerase, so these polymerases basically are, as I said, 5 types. So polymerase 1, it involves in the removal of the primers. So it involves in the removal of the primers. So the second one is the replication. So polymerase 3, which participates in the replication of the DNA. So the another one is the repair. So this repair is this coming to this repair. So both that polymerase 1 and polymerase 3 can remove the nucleotides from the 3' end, from the 3' end. But only the polymerase 1, so the polymerase 1 can remove the DNA even from the 5' to 3' end. So that's why, so we say that no, it has both 5', 3' exonuclease activity and also the 3' exonuclease activity. When DNA is removed in the from the 3' direction, so we say it is a repair. So when DNA is removed from 5' to 3' direction, so it is called the removal of RNA primer. So DNA polymerase 1 has this function of removing the 5', 3' end, 5', 3' end, also the 3' and 5' end. That means that the DNA polymerase 1 can perform both the repair function and the removal function. And coming to the DNA polymerase 3, so which has repair function and also the replication function, but it doesn't have the removal function. So then coming to the polymerase 2, so what is the function of the polymerase 2? So this polymerase 2, so it only involves in the repair. It doesn't involve in the removal of the primers, or it doesn't involve in the replication of DNA. It involves on the repair. So when bases, for example, thymine, when thymine bases are present, when thymine when these thymine nucleotides are present in a DNA strand, so this thymine nucleotides, when they're present side by side, so when these thymine are exposed to UV light, so this thymine forms the thymine dimer. So when thymine dimers are formed in a DNA, so that these polymerase enzymes, for example polymerase 3, when while synthesizing the DNA, when it encounters these thymine dimers on the DNA, so this polymerase 3 cannot proceed further, so that the polymerase 2 now completes the DNA replication in place of the polymerase 3, where the DNA forms the thymine dimers. So this thymine dimers region is completely synthesized by the particularly this particular polymerase 2. So then polymerase 4 and polymerase 5, see this polymerase 4 and polymerase 5, so these polymerase 4 and 5, they also participate in the replication of the DNA. So this is how this polymerase enzymes which participates in the removal of the primers, also this they involve in the replication of DNA, also they repair the DNA while synthesizing daughter DNA. Okay, so coming to the structure, so coming to this structure, so this polymerase 3, so it has some subunits. So we know that even the hemoglobin, where subunits are there, like 2 alpha subunits and 2 beta subunits are there. So like that in a polymerase 3 also, so in this core protein, so this core protein is formed by the subunits. So the subunits are called here alpha subunit, epsilon subunit and theta subunit. This alpha subunit has the polymerase activity. Okay, then this epsilon unit has the exonuclease activity, and this theta subunit, which is important for the stabilization of this epsilon subunit. So these three subunits together, they form the core region of this polymerase enzyme. So when we look into the holoenzyme, so this is the core region only. So this core region is formed by three subunits, alpha, epsilon and this theta. So when we look into the other subunits of this polymerase 3, so tau proteins are there. So these tau proteins are very important. So these tau proteins, they keep the polymerases, that means it keeps the two polymerases together, so that each polymerase can synthesize the respective DNA strands, because the DNA contains two strands. So that's why, so two polymerases are required to synthesize the daughter DNAs from the two template DNAs. Therefore the two polymerase enzymes are connected by this tau proteins. So this tau proteins helps to keep the two polymerases together. Okay, then there are proteins like delta prime, delta, gamma proteins are there. So these gamma, delta prime, delta proteins are called clamp loading proteins, clamp loaders they are called. So these clamp loaders we can see here, these clamp loaders we call beta sliding clamps. So these beta subunits, they forms this sliding clamps. So the function of these sliding clamps is that, so they bind to the DNA strands, so that you can see that these sliding clamps, they are ring like structures. So when this clamp opens, so that DNA strand, so this clamp protein enters, encircles the that complete DNA strand. So by opening this, so it encircles the DNA strand, and then again it closes, so that this clamp fixes onto DNA strands, single stranded DNA strands. So since these clamps are present on the this DNA strands, now these particular polymerase enzymes, they bind to this particular clamp, so that polymerase enzymes, they don't dissociate immediately once they are loaded onto the DNA strand. That's why their processivity is increased by this sliding clamps. So processivity is very important. The polymerases have, especially this polymerase 3 has high processivity because of these sliding clamps only. So the sliding clamps keeps these polymerases for a long time on the DNA strands during the replication. So these subunits are present in this polymerase 3 enzyme. So all these subunits with this core enzyme, they forms the whole enzyme. That is very important. So here also we can see that beta clamps are there. So these beta clamps are loaded by this gamma protein and proteins like delta prime protein is there. Also the chi and psi proteins are there. These chi and psi proteins, they are helpful for keeping these gamma subunits together, or they stabilize this gamma complex. So the gamma complex is stabilized by with the help of the chi and psi proteins. So tau proteins are there. The tau proteins, they keep these polymerases together, and these clamp proteins are very important. So they increase the processivity of these polymerase enzymes. So this is about the polymerase enzyme. So when we discuss about the polymerase enzyme, we have to understand that. So the exonuclease activity and polymerase activity. So both these properties are present with the same enzyme, that is called a polymerase enzyme. So it's not like that exonuclease is a one separate enzyme, and this polymerase is a separate enzyme or the proteins. So there are subunits of this core complex, so where they have this both exonuclease activity and the polymerase activity. So basically these polymerases, they perform three types of functions. One is the removal function, that means removal of primer. Another one is the replication function, replication of the parental DNA by synthesizing daughter DNAs. The other one is the repairing of DNA. So during synthesis of daughter DNA, sometimes a wrong nucleotide may be incorporated in the daughter DNA. So such wrong nucleotides are removed by this polymerase enzyme. So thank you for watching this video.